And we know that metabolomics is a comprehensive or exhaustive profiling of these metabolites, and it's the final readouts of metabolism. And so it's very highly correlated with physiological phenomenon. And so when we try to do food quality evaluation using metabolomics, we could do so by determining the ratio of chemical constituents. We could also use metabolomics to seek for a specific marker related to a particular physiological phenomenon. And then we could also do a global analysis of these food in order for us to understand the differences of metabolites present in a variety of food samples. And I would like to bring your attention to our nation, Indonesia. Even though I've been in Japan for 17 years, many of my research is still about Indonesia because it's really close to my heart. And I would like metabolomics to actually contribute in improving the quality of Indonesian bioproducts. So we know that our nation is very rich in bioresource and we have abundant food production, but still crop loss often occurs. Many of these are caused by either disease in plant and aquaculture product, poor post-harvest management, so the food products already spoiled even before it reached the consumer, or because it's in very low quality, so it cannot be sold. So it's basically just finally be given to feed for animals. Instead of if it's processed in a very high quality, actually it could be sold to consumers or even go for export. So how metabolomics can tackle these issues? We could assist the investigation on the cause of disease. We could also aid systematic and effective post-harvest management, and we also could improve the quality by using metabolome information to assist quality improvement. So I would like to remind you again of all of the different metabolites that are covered in metabolome. And also here I already mentioned that it's a combination of bioscience, analytical chemistry, and application. And here I would like to talk about the concept of food metabolomics. So for food metabolomics, the concept is actually very simple. How to evaluate the quality of food. If we would like to improve food quality, we first have to have a very clear criteria on how we determine food quality. For example, I like Indomie, soto ayam, and you like, Indomie, maybe kari ayam. So we both have different preference. What we consider good or delicious is different. Maybe I like flavorful and spicy, and you like salty or sweet. Then we cannot agree on what is good quality because there's a bias. So that is called preference, and we could not use that kind of preference to correlate with metabolome. What we can do and to do the correlation with metabolome, if you and I agree on the same criteria and we have the same standard, and it's determined by something that is quantitative and can be measured. For example, okay, we agree that in this food sample, the bitterness is level 5. And the bitterness is determined, for example, by measuring different concentration of caffeine solution. And then we agree, okay, this level is 1, this level is 5, this level is 10. Then we standardize our measurement. And then when we were given a sample, we could measure the bitterness. That is descriptive and also quantitative. So if we have that kind of analysis, then bitterness in food can be correlated with a particular metabolite and their concentration. But if it's just delicious, we cannot, it's not specific and quantitative, and it's not describing the food enough. It's just delicious. Delicious can be many different things. Delicious, you and I can both agree on different terms of delicious. But if it's bitterness, sweetness, acidity, for example, astringency, chewiness, and texture, if it's described in a quantitative manner, then this can be a measure of food function. So later on, this kind of food function that is determined by usually trained or semi-trained panelists can be a way to measure the quality evaluation. And the same food samples are subjected to different kind of instrument analysis and analyze these components in a quantitative manner. And then using informatics approach, we could create a model where we could correlate these food functions with these food components. Later on, I could later explain, for example, this food is more salty compared to this one. Why? Because it contains maybe this specific amino acid that gives salt flavor. Or, for example, this kind of food is more bitter. Why? Because, for example, in this sample, it has higher caffeine, and caffeine gives bitter taste. Things like this. Then you could also measure quality. For example, quality is determined. This food has high quality if it's blah, 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 blah. Then if quality can be determined in a non-biased quantitative manner, you could also do the same by correlating this food quality with particular component. Then later on, you could explain the food having high quality always has increasingly higher component of something, something. Then if you could find this quality marker, you could use this quality marker later on to improve in your food, either in your food processing, or you could find breeds or cultivars or particular type of food having this high concentration of this marker, because that will then correlate to having high quality. So this is a typical workflow in food metabolomics. The most commonly used approach is just to determine the differences between two different kind of class. So then afterwards, what makes these classes different? Then you have a discriminant marker to tell the difference between two types of food. And you could also have informative analysis, for example, elucidation of pathways involved in pathogen infection. This is one of the examples I mentioned, how metabolomics can assist in investigating the cause of disease, for example, like what happens after pathogen infect a particular plant. And then we could see the changes in the metabolism or the self-defense response. And then this could give us an information on then how we could manage to detect these infection early, or to develop like a biocontrol agent to prevent these plant infection. Or we could also use it as a predictive way to predict food quality, because we know that if we use sensory analysis by humans, that is the first step. Of course, we want to do that. It's important to construct the model. But afterwards, once we have the model and we already know what kind of metabolite is important for particular attribute, then we could complement the use of human sensory analysis with metabolome analysis, and then we could increase the throughput. Because if we use human samples, humans have very limited capacity to measure how many different samples, because after some time, your tongue will be numb, and we call it fatigue, sensory fatigue. Humans will not have the ability to measure many different kind of food at once. Afterwards, it just doesn't taste different anymore. Then you will have the food fatigue. Then afterwards, it's very good if you have a complementary method to screen first in high throughput, and then only the very, very important ones is subjected to human sensory analysis for final validation. So metabolomics approach can be applied to many different kinds of food products. This also applies to agricultural products or herbal medicine, of course. Natural food product, it can be agriculture or aquaculture product, processed or fermented food product. And in our laboratory, we have many different kinds of samples, like fermented food, aquaculture samples, as well as agriculture samples. And the application is quite wide. We do the component analysis for quality improvement, authenticity assessment, and diet monitoring. So first, I would like to talk about the application of metabolomics to propose the quality marker for specialty coffee, because this is about quality assessment. So this is one way that we could complement the present quality assessment by proposing a new quality marker for specialty coffee. I'm not sure if everyone is familiar here with specialty coffee, but it's just not your ordinary coffee. It's a little bit expensive, and because usually it comes from a single origin, and it's maintained really well. And usually we have a very clear criteria on determining whether a coffee sample can be classified into specialty grade or just a normal coffee. So the most common method to use in this cupping test analysis to determine the coffee quality is using a trained panelist. Usually it has to have a license, and we call it the license Q grader for Arabica coffee. And the licensed panelists usually consist of a team of 5 to 10 people. And then afterwards, we measure 10 different attributes. The attributes are written here. And each attribute will have an intensity of maximum to 0 to maximum of 10. Then the full points will be 10 attributes times 10. If it's full score, then 100. But if the cumulative point is higher than 80, it's considered as specialty grade. Then specialty coffee gained a lot of attention in recent years. I think everyone knows nowadays, whenever we go to very nice coffee shops, they always offer like, would you like to have today's coffee is Aceh coffee or Mandailing coffee. We are a nation that is really, really good in producing very nice coffee. And we have a very big variety of specialty coffee from different regions. And each of these coffee, they might have the same points, maybe 82, 82.5, but they may not have the same flavor. Because as I mentioned, the quality of coffee is just determined by the sum of everything. But the individual score may not be different. It may be different for different types of coffee that we have. So, well, what I would like to know, is there a commonality every time cupping score increase? Even though the coffee I know is very different, coffee from Aceh and coffee from Papua will be very different in terms of their aroma, the acidity, the flavor. But is there something in common that is always increased when cupping score increase? So this one, the objective is to propose the quality marker for specialty coffee only. So we collected various kinds of specialty coffee. Actually, we received 120 samples from Specialty Coffee of Association of Indonesia. At that time, ICCRI was the host for organizing the coffee competition, national coffee competition for specialty coffee. And they give us many samples, and we selected 40 among the samples. But we first select 10, having a representative from 1 to 10. The lowest having the range of 80.25. This is like a borderline specialty coffee, just a little bit higher than 80. And the highest in that year was 86.75, which is really, really high. Then you could see here from number 1 to number 10, this is in the spider web, you could see that some of them have quite similar characteristic, how these scores are graded, but some of them have quite different score also in terms of, for example, in the balance or like that. So the way to do this analysis is by using GCMS-based metabolic profiling, and the hydrophilic low molecular weight compounds were the target in our GCMS analysis. We do a derivatization so that we could measure non-volatiles. Usually GCMS is good for volatiles, but we use derivatization methods, so we could also use non-volatiles like sugars and amino acids. We also have organic acids and other metabolites as shown here. We annotated, including the unknown compounds, we detected 97 metabolites. And then these 97 metabolites are used as the explanatory variable to make our regression model. And the response variable, which is the variable that we would like to explain, is the cupping score. So here we have this orthogonal projection to latent structure, also called OPLS model regression. This is a multivariate analysis method to find the relationship between Y and X, and it will maximize the correlation between Y and X So we would like to explain the cupping score and what kind of metabolite has the tightest correlation with the cupping score. So the OPLS introduces the latent variable called P that has a maximal correlation with the Y. In this case, Y is the cupping score, and cupping score is used to define coffee quality. Then here we have these metabolites. The explanatory variable is used to as variables to find the correlation with Y. Then after the model is constructed, if we could have a good model from these 10 samples, we will have this type of two different terms. First of all, we call it the VIP score. Compounds of which VIP are greater than 1 are considered to be important for the model. So this is how we find whether some particular metabolite contributes to coffee cupping score. And if the regression coefficient is positive, that means that these compounds positively contribute or positively correlated to the cupping score. And if it's negative, then it's negatively correlated to the cupping score. So what we want to find is something with positive score and also important for the model. That means as cupping score increase, this metabolite also increase. But if you also would like to find like bad metabolites, the one that cause defect, you could also screen for the metabolites having negative correlation. In that case, you would like to prevent these negatively contributing metabolites in your sample. We make a model using the final score as the Y variable, but we also use the individual score from each of the attributes. And we don't use all because some of the attributes, for example, I forgot what, but some of the attributes has the same score for all of them. So then it cannot be used to be a model. Among the 10 samples, they all have the same score. So we use only the attributes that have a range of scores in order for us to make a model. Then what we found very interesting is that the model was shown to have very good performance, shown by the R square and Q square. And what's interesting is for the final score, we found that galactinol in many of the attributes contribute positively in many of our models. So galactinol was proposed as a quality marker based on the prediction model of 7 attributes, including the final score. So we found a very high contribution and positive coefficient for galactinol. And we also measure in our samples, and indeed, the concentration of galactinol increased proportionally. And we also attempted to do a quantitative analysis of galactinol. We found that there is at least 10 to 15 times higher concentration of galactinol compared to the one that has from 80 to 86.75 cupping score. The highest and the lowest one have quite different concentration of galactinol. We also found that sucrose have a positive coefficient and also high VIP. And sucrose has also been reported to have a correlation with coffee flavor in the previous experiment. So sucrose has already been reported, and we validated that we found the same compound was also detected as a positively contributing metabolite for flavor. So that is one of the way we could propose, then we could design the experiment in order for us to improve the coffee quality later on, perhaps by targeting this compound galactinol in coffee. Then I would like to show you a little bit. This is a flashback going back to 2013 when I published the first report on the coffee authentication of Kopi Luwak. And then it was displayed in several newspapers, actually, in Science Magazine and The Economist, because it was the authentication method of the world's most expensive coffee. And that was the starting point of, okay, now we know what makes kopi luwak different with normal kopi. Then we continued on also to discriminate Indonesian specialty coffee, not only coming from kopi luwak, but also specialty coffee in general, coming from many different kind of area. So we also collaborate with ICCRI here in Pusat Koka Jember to compare the metabolite profile of coffee samples coming from different regions in Indonesia. Here you could see that we have samples from Java, from Sulawesi, from Sumatra, and we have Robusta and Arabica in green or roasted state. And when we compare these factors simultaneously, you could see that we have three different variables. We have the roasting process, species, and geographical origin. And then we analyze all of these samples together in order for us to understand which factor is most influential in, which factor is more influential in separating the samples and which factor gives the highest variance among our samples. And here we found in principal component 1, the order of factors of classification was determined. The first one, roasting process, greatly changed the metabolite profile of coffee so much, of course, because it's heated to 200, 300 degrees, and so many loss of metabolites. So then green and roasted metabolites, coffee metabolites are, of course, quite different. So we could see the influence of roasting. For example, in green coffee, we have higher alanine and GABA, but after roasting, we generate many different kind of acids. In principal component 2, the second factor, we could see that species, the Arabica and Robusta, give the second biggest influence in the separation. And then we cannot see geographical origin. It's just Arabica and Robusta and green and roasted. So the differences of metabolites due to roasting process and species also match previous reports, but we would like to see the differences between samples coming from different regions. And therefore, we need to remove the influential factor in our analysis by analyzing only the green Arabica coffee beans. So in the next analysis, we limit the analysis to make the roasting condition the same. So all of them are green beans. And we analyze the Arabica and the Robusta data independently. And then we expand the sample of coming from different regions, from 10 different geographical origins in Indonesia. Then it's very interesting that we could see very clear separation of samples based on their geographical origin. And the samples which have close geographical origin are not always plotted closely in score plot. You could see that for Java sample here, Java sample actually one comes from Andung Sari and one comes from Malang. So it's different region in Java, but they are clustered still closely to each other. We have samples also from different places in Flores, but they are clustered closely to each other. 2 samples from Sumatra, for example, Mandeling and Aceh. Gayo is also clustered closely to each other. So samples that are coming from the same island or are clustered closely to each other. And somehow we could see like there's a middle part of Indonesia, an eastern part of Indonesia, or western part of Indonesia somehow is also clustered together. So that was published in Metabolomics Journal in 2019. And we would like to further use the kind of information we collected from analysis of different kind of specialty coffee from all over Indonesia. We combine our know-how also on civet coffee and how the bacterial consortium in civet gut might be influential in changing the metabolite profile of coffee. So we know that in post-harvest process of coffee here in the wash process, it also involves a very important step here where fermentation takes place. The wash process involves natural fermentation after pulping process. And when we when we compare the luwak samples and non-luwak samples, we know that the digestion of luwak is quite influential in changing the metabolite profile of luwak coffee, but the change is not as significant that it doesn't change the characteristic of each region significantly. So, for example, kopi luwak from Aceh will still look like non-luwak coffee from Aceh. It doesn't suddenly change to something like Bali. So the original characteristics still remain, but the change is quite mild in a way that it could be used to enhance the quality of coffee. So from that point, we made a strategic decision to isolate several isolates from civet cat excrement. And we would like to develop a method for second fermentation where we employ, first we screen for enzymes having amylolytic activity, cellulolytic activity, pectinolytic activity, and proteolytic activity from civet coffee. But unfortunately, we couldn't find cellulolytic activity from kopi luwak, which is actually essential for green coffee analysis to process the cellulose from the green coffee beans. So we use a different kind of strain from our samples that we isolated from ubi cilembu that has very high cellulolytic activity. Then we optimize these three inoculum, different ratio. We also optimize different hours of fermentation, 4, 8, 12 hours. And then we subject these combination of different ratio into 3 different kinds of coffee coming from 3 different regions. One is from Java, one is from Sumatra, one is from Bali. And you could see here that the clustering can still be seen. Even if it's after fermentation, the cluster is still very closely clustered with the original non-fermented coffee beans, but then it becomes different. So if you analyze the samples individually, then you could see the fermentation A, for example, I4B means that it's annotated for 4 hours in coffee coffee I. And this one, B is just optimize the inoculum ratio, different kind of inoculum ratio. So fermentation A and fermentation B has already been selected as the best combination of the inoculum and the hours of fermentation. And these differences in the inoculum ratio and the hours of fermentation also give rise to different characteristics of coffee after fermentation. Then, in short, we subject this to different kinds of coffee, as I mentioned before. And this is the description. For example, for A, B, for B, for C is duration of fermentation is 4 hours. And then we have the ratio of isolate is also different. One is more dominant in the cellulolytic bacteria. One is more dominant in the amylolytic bacteria and things like that. And then we have a very surprising and exciting result that we were able to increase the cupping score from 81 to 85.3 by doing the second fermentation of coffee beans. And we have registered this method as a registered patent in Indonesia in 2019. And the papers were published, the paper was published in Metabolomics Journal by Springer in 2020, last year. So I think, do I still have time to the moderator?
Yes, you still have the time.
Okay. So, so that is the story about coffee and how we could use the metabolome information to know the characteristics of different coffee and also how to improve the coffee and how to do the quality assessment of coffee, not only using sensory analysis, but hopefully using the quality marker that we proposed. And then we also analyzed Tempeh. This is in collaboration with Professor Made Astawan and Professor Pinkan Adityawati. Professor Made is also a professor in IPB. And we have different kinds of tempeh from made using traditional methods, semi-modern and modern, and coming from different regions. And then we could see very beautifully that the samples of tempeh coming from Bogor and Bandung are actually quite similar. whereas the samples of tempeh coming from Central and East Java are clustered separately. Then we could also see here the differences between tempeh that are produced in modernized way, traditional way, and what kind of metabolites are important to separate these tempeh that are produced in different ways. And we also subject metabolomics for optimizing the different post-harvest treatment in mangosteen. This is also a collaboration with Professor Sobir in the PKHT in ITB. And not only about mangosteen, we also do research on many different kind of tropical fruits, including mango, mangosteen, pineapple, and coconut. And here we could see here that this is a very interesting physiological changes that happen during ripening. You could see that when fruit ripens, the color changes, aroma changes, and usually it becomes sweeter, and also it becomes softer and easier to open. So these changes are the result of dynamic processes of molecular and biochemical changes, and we would like to monitor the metabolite changes that happen during the ripening of mangosteen. So why mangosteen? Because it was, it is still one of the most exported fruit compared to other tropical fruits that are produced in Indonesia. But there's a very big challenge to keep mangosteen shelf life, because after it is taken out from the tree, it just ripes to the following stage for 2 days in room temperature, and the color changes quite rapidly. Then we would like to know the effect of post-harvest mangosteen technology. At first, in the first study, we already know what changes during the course of ripening. And then the second one, we would like to use several different kind of post-harvest technology and see how it affects the mangosteen metabolite profile. So here, this is the work of one of my PhD students before, who is an alumni, also from Indonesia. And she investigate the changes of mangosteen after being subjected to low temperature and also to stress inducer like methyl jasmonate or salicylic acid. And the sponge and water is just as a control, because methyl jasmonate and salicylic acid uses water to dissolve and then use the sponge. So this is just used as a control. And from all of these, actually what we use this because according to the previous reports in the literature, these are all low temperature methyl jasmonate and salicylic acid was found to be effective in prolonging mangosteen shelf life. But in our analysis, we could only reproduce that low temperature is effective. Then here we compare the progression of ripening between the samples that are kept in room temperature and the samples that are kept in low temperature. And then you could see in the blue colored one, the progression along PC1 from the negative axis to positive axis did not happen the same way as the red colored one. In the red colored one, where the ripening progresses normally, you could see a very nice change from the samples in which along the ripening stage progresses, fructose, glucuronate, galactose, and galacturonic acid actually increase over time. And this fructose and galactose is involved in making the mangosteen sweeter, and the glucuronate and galacturonic acid is important to make the peel softer, which is one of the most important characteristics that happens during ripening. But in the samples that are kept in low temperature, you could see that even the day 10 samples, the characteristic is quite similar compared to like day 5 of the normal ripening in room temperature. That means room temperature is delaying the mangosteen ripening process in peel part by perhaps making the conversion or the synthesis of glucose and galacturonic acid to be much slower in low temperature. And this polysaccharides breakdown activity in low temperature has a lower relative intensity compared to room temperature storage. But what's interesting is that was the data we take from peel. But when we open the fruit and we analyze the flesh, actually room temperature and low temperature both do not have any significant difference. So what we could see, actually, low temperature mangosteen is effective in delaying the ripening somehow, as we could see very clearly in the peel part. But the characteristic of the metabolite profile changes in the flesh part that we consume is actually not significant. So we could say that there are some differences between the characteristic of metabolites that are, for example, the aroma precursor compound during the ripening process in low temperature is much decreased compared to the room temperature. But since we couldn't find any trend in the flesh part, the conclusion of this study is the present way of prolonging mangosteen shelf life is effective to prevent the color change from the outside appearance of the peel, but it's not so effective in actually delaying the changes in the metabolite profile of the peel part. So we still have to find effective ways to prolong mangosteen shelf life. So similar work was also done using non-climateric fruits in pineapple, where we could find different changes in metabolites during pineapple ripening. This is done by one of my PhD students, also coming from Indonesia. And among everything, I thought that we have such a rich experience of analyzing many different kinds of tropical bioproducts from Indonesia. So what's next? What's next is I thought that maybe as an academia, we also have the responsibility that all of these results can actually be translated into something that could improve the quality of life and improve the livelihood of our farmers. And so to do that, my family and I also established a company in Indonesia for export and import. And mostly we do export of many different kind of commodities, such as our spices, herbs, and fruits, as well as fermented food like tempeh, in order for us to be able to increase the quality and train the farmers. And then these high quality produce can be exported to markets to increase their value. So this is one of the ways that we could continuously translate our research to be not just about papers publishing, but actually doing real applications to the field and actually translate that into improvement of the quality of our produce, and then improve the quality in order to meet the global standards, and then be able to export our high quality commodities from Indonesia. So with that, I would like to close my talk by summarizing that in metabolomics is quite a useful technology and can be used in many different kinds of application area. And then it's very important for us to consider how we should support this post-harvest development strategy, controlling the diseases, and improving the quality of our products, because it's very important to strengthen our nation's food security. So with that, I would like to close this talk by thanking all of the students, my really wonderful students who have contributed to many of the research that I described today. And here is a picture of our students in Batik Day last year. And these are some statistics of our students. In my team, we have 19 students, 10 PhD students, 5 master students, 4 bachelor students. And we have students coming from different nationality. From Indonesia, we have 7 students, one of the highest number in Osaka University in one lab. And of course, very much welcome. If there's any questions or you want to apply to our group, you could email me at this email address. And with that, I would like to close my talk by thanking everyone for your kind attention, and very welcome to have a discussion afterwards. So I would like to return my talk back to the moderator. Thank you very much.
Yeah, thank you so much to Dr. Sastia. It's another interesting topic. Now we open a discussion session again. Actually, there is one question in the column chat from Dede Sukandar from UIN. I want to question to Dr. Sastia. There are several multivariate analyses, including PCA, PLS, OPLS, and OPLS-DA. How are they different from each other? Thank you for your answers.
Okay, thank you very much. So actually, it's a very nice question, and the principal component analysis is usually used to as an unsupervised analysis, means that we just analyze the samples as they are, and then if we would like to do just a quick glimpse of what the data looks like and how the samples are clustered with each other and what are the factors that might be important in the discrimination. PLS and OPLS is a regression model where we usually use this to find correlation between a particular attribute or phenotype that we assign, and it has to be quantitative. And usually we determine the score or the value beforehand for this making this regression model. And this is the point if we are interested to find the correlation between metabolites with a particular phenotypic characteristics or attribute. And then this is how we could maximize the correlation. And this is a supervised analysis where we could say, okay, the model, please find metabolites that is most correlated with these values. And these values are a definition of a particular characteristic that we're interested on. And for OPLS-DA, is actually also to tell the difference between one class and another class. So DA is discriminant analysis. For PCA, we cannot know sometimes if, for example, we want to tell the difference between copy luwak and non-copy luwak. But then when we do the PCA, because it's unsupervised analysis and it's non-biased, we cannot see the differences between copy luwak and copy luwak. What we could see is just the differences between coffee from different origin, regardless whether it is digested with luwak or not. Then we cannot achieve our study objective, which is to find the differences between copy luwak and luwak. In such case, we need to do this supervised analysis where we tell the multivariate analysis, okay, class 1 is copy luwak samples. Class 2 is non-copy luwak samples. Please tell us the difference between these two samples. So this is supervised analysis, and of course, it's quite different principle with how PCA is done, because the objective is already to find the differences between two different classes. So it really depends on the objective of what you would like to achieve from your analysis. And at times, it depends also on your biological question. If you would like to find specific metabolites correlated with a specific phenotype, it's always good. And you have a range of these phenotypes having different values in different samples. PLS or OPLS is quite suitable. But if your objective is to do the discrimination between the two samples and you cannot achieve that by doing a PCA, then you use this supervised OPLS-DA. And the very common ones to do first is to know the data structure is to do PCA. Okay, I hope that answers your question.
Okay, thank you so much for the answer, Doctor. We still have a time, maybe one question more before we end the discussion session, because due to limitation of time. Please, to the students, you can ask Dr. Sastia directly. Maybe one more question. While we wait the question from the students, maybe I would like to ask, is it true if the metabolomic research is always yields the sample biomarkers or something, ma'am?
The sample biomarker, it depends if our study is to find biomarkers like metabolite markers, then if that is the objective, then we hope to find it, but not always. It's not always successful because sometimes there's no clear trend. For example, if we hope that during the course of ripening, something increase along with ripening, right? Linearly. But sometimes it doesn't. Like the metabolites are just like this, or you cannot find a very strong metabolite that increases over time. So you just find like metabolite changes globally, but not a particular metabolite strongly correlate with some attributes, or not always that a particular metabolite linearly increase along with time course. So the answer is not always we find marker.
Okay.
Yeah, especially if the study that we would like to do is also very complex. For example, like we want to find the metabolite marker for cancer. Maybe the same person can get cancer, but it's very, very different depending on the metabolites, the diet, the environment. So there's not one single marker that can explain the progression of cancer, for example. It's because so many different complexity. Yeah.
Okay, thank you so much for the answer, Dr. Sastia. Due to limitation of time, maybe we end the discussion session here. It's a great pleasure for us to have you here as a speaker. Maybe to the student who still have a question, maybe you can write the question on the chat column so the committee can note your question and send it to Dr. Sastia directly via email. Once again, thank you so much, Dr. Sastia, for the lecture and the answer.
Yes, thank you very much. It's my pleasure. Thank you, everyone.
Okay, so